If you look at examples of living things around you, at a tree, a mushroom, or a plant, they all seem worlds apart and sometimes really diverse. And on top of that, there's an entire world of microorganisms that's hidden from our eyes. Yet, all life shares a common ancestor. It turns out we can actually learn a lot about the evolutionary history of life by studying the differences in their cellular architecture or structure. So, all life can be categorized into three broad groups called the three domains of life. This is a large taxonomic grouping. From there, any given organism gets placed into more specific groups, and the tree of life continues to branch all the way down to the species level. First, we have the bacteria. They're really tiny, microscopic. They are single celled and distinguished by the lack of membrane bound nucleus or other membrane bound organelles. The second domain of life is the archaea. Haven't heard of them? That's because they are the least studied. Actually, scientists used to believe that archaea and bacteria were all part of the same domain of life, and they were called the prokaryotes. That was just because they seemed to be structurally similar on the surface. Archaea are also small, single celled, and contain no membrane bound organelles, just like the bacteria. However, on closer inspection, the archaea share a lot of interesting similarities with Eukarya, our third and final domain of life, that we're soon going to dive in more into. So that means that the prokaryotic designation doesn't really make sense. While archaea look similar to bacteria, they have unique membrane lipids, for an example, or other transcription machinery that's more similar to eukarya than bacteria. So just know that when you hear this term prokaryotic, it's often just referring to a cellular structure that both the bacteria and archaea share, but it's not a real taxonomic grouping of life, and it just implies the wrong evolutionary history. So let's take one more second to talk about the eukarya. They are defined by compartmentalization within the cell. That means they have a membrane bound nucleus where their DNA is found, and they have other membrane bound organelles. Eukaryotic organisms can be microscopic, like algae, amoeba, or yeast. And then others are multicellular, like plants and animals, and can be, therefore, macroscopic. How did we get from a simple prokaryotic cell to the complex eukaryotic cell? So it wasn't just a slow evolutionary change over time via mutation. There was actually what seems to be a merger event, and that's the coolest plot twist. So let's explain how this came to be. To describe this pivotal moment, we need to look at the relationship between the host, likely an archaea in this story, and the guest, the bacterium. So recent genomic evidence suggests that the host was likely an Asgard archaea. Unlike typical bacteria, these organisms had eukaryotic signature proteins that allowed them to manipulate their cell membranes to create protrusions or tentacles. The guest was likely an alpha proteobacterium. This was a small gram-negative bacterium that had mastered oxidative phosphorylation. Big word, but it basically means the ability to use oxygen to produce ATP. ATP is the energy currency of the cell, so super important. In a world becoming increasingly oxygenated, this was a superpower. Now, this process didn't happen overnight. It likely followed a couple of steps. So, first we have proximity and feeding. The Archaean host likely lived in close proximity to the bacteria, so syntropy, they might have had some sort of relationship, and perhaps they were feeding on the waste products that bacteria produced. At some point, we're seeing a sort of tentacle wrap. So, using its cytoskeleton, the archaean extended its membrane around the bacterium. This maybe wasn't even necessarily like a predatory gulp. It may have started as an accidental entrapment between the two organisms when they were sharing nutrients. Eventually, though, we see some sort of phagocytic event. This means that the bacterium ends up fully enclosed within a vesicle inside of the host. And this is something that we see happen with eukaryotic cells today, this phagocytic activity. An interesting question, though, is why wasn't it digested? Usually, an engulfed cell is dissolved by enzymes or food, but in this case, we think that two factors at least might have helped prevent this. So, the first is mutual benefit. The bacterium provided the host with a massive surge of energy, aka ATP, in an oxygen rich environment that was otherwise toxic or inefficient to the archaean. We also likely saw horizontal gene transfer. This means that over time, the bacterium was sort of like leaking its DNA into the host cytoplasm, and the host is integrating those genes into its own genome, its own nuclear DNA. eventually taking control of the bacterium's replication. So a key distinction here is that this transformed the relationship between these organisms from facultative, meaning they can live apart, to obligate, meaning they can no longer survive without each other. The bacterium was no longer a separate organism, and over time, we have the evolution of this bacterium as an organelle. Again, it's thought that this evolutionary process solved what can be called the energy crisis of the cell. Bacterial and archaeal cells, called, the prokaryotic cells, are constrained in size because all of their energy production takes place on the inner cell membrane. By having internal power plants, the mitochondria in eukaryotic cells, well, these eukaryotic cells could grow much larger and maintain a more complex genome, think DNA and genetic code, more so than any prokaryote before it. It's thought that this provided the raw energy needed to fuel the evolution of multicellularity, think animals and plants that we see today. Perhaps the next logical question is, do we have any evidence that this actually happened? And short answer, yes, we do. I'll give you a couple examples. First up is the membranes. Mitochondria have a double membrane. While we traditionally thought that the outer membrane came from the host's vesicle, modern proteomic evidence suggests that it might actually be the preserved outer membrane of the original bacterial ancestor, which was gram-negative, meaning that it had two membranes. Additionally, there's something funky that goes on with antibiotics. Mitochondria are susceptible to certain antibiotics that target bacteria, suggesting their bacterial origins. Additionally, mitochondria produce via a process that looks a lot like binary fission. This is what we see in bacterial cells. When the mitochondria reproduce, they do so independently of the eukaryotic cell's division process, like mitosis. And actually, if you remove all mitochondria from a cell, the cell can't make new ones from scratch. Finally, we can take a look at DNA and ribosomes. Mitochondria have their own circular DNA called mitochondrial DNA, and it's separate from the nuclear DNA, or you can think main chromosome or chromosomes of the eukaryotic cell. Perhaps not coincidentally, most bacteria have one singular circular chromosome. On top of that, mitochondria have their own ribosomes, which are 70S in size. This is the same that you find in bacteria, rather than 80S, which is the size that you find in the eukaryotic cytoplasm. And in case you forgot, ribosomes are an organelle, though they're not membrane bound. And they're found across the entire tree of life. It's super interesting then that mitochondria, which are inside of eukaryotic cells, contain their own DNA and their own distinct ribosomes. And we must clarify that it wasn't just the mitochondria. There's also really strong evidence that the chloroplast, an organelle involved in energy mechanics and photosynthesis in taxonomic groups like the plants and the algae, also evolved through an endosymbiotic event similar to this one. Ultimately, the emergence of the eukaryotic cell was not just a slow accumulation of mutations, but also a revolutionary merger that redefined the potential of life. By transitioning from a simple, single compartment existence to a sophisticated partnership through endosymbiosis, these ancestral cells overcame the energy constraints that still limit the domains of bacteria and archaea, even today. And hey, that's not to say that one domain of life is better than the other. Remember that anything alive today is evolutionarily a fit strategy, and it's one that works. However, the emergence of the Eukarya through this endosymbiotic event really did pave the way for a massive diversity of larger multicellular life like humans, animals, and the plants that we see today.